Hello everyone, and welcome to today's Lab Tools webinar. I'm Nikki Spahich, Associate Science Editor for The Scientist, and I will be moderating our discussion. Today, our speaker, Dr. Nicholas Hund, will discuss a new mass photometry method that characterizes molecules in their native state on supported lipid bilayers. During the live Q&A, he will be joined by Tamara Herrmann and Frederick Steyert from the laboratory of Petra Schwille at the Max Planck Institute of Biochemistry. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We'll try to address as many of these questions as we can during our Q&A session. The webinar will be archived on the Scientist website. We will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. The pioneer of mass photometry, Refine produces a disruptive new generation of analytical instruments that measure the mass of individual molecules directly in solution. Mass photometry transforms the ability to characterize the composition, structure, and dynamics of biomolecules, revealing the true behavior of molecules in their native environment. Refine aims to accelerate research and enable discoveries by sharing mass photometry with the global scientific community. Our sponsor has provided us with some helpful resources related to today's webinar. We have posted these in our handout section, located on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our speaker. Nicholas Hund received his PhD in biochemistry in 2015 while working in Dietmar Manstein's laboratory at Hanover Medical School, Germany, where he characterized non-muscle myosins using fluorescence microscopy and optical tweezers. In 2016, he joined Philip Kokura's laboratory at the University of Oxford in the UK as a postdoctoral fellow, where he studied actin nucleation and polymerization using interferomic scattering microscopy, or ISCAT, and co-developed mass photometry. Hund is currently a postdoctoral fellow in Claudia Feigel's laboratory at Ludwig Maximilians University of Munich, Germany, where he explores new applications for ISCAT and mass photometry. Let's just make sure your slides are running. Okay, Dr. Hund, take it away.
Thank you for the kind introduction. And I'm also thanking Refine for sponsoring the webinar. And thank you all for joining the webinar. I'm very excited to share some of our most recent work about how to use mass photometry to track membrane proteins with you. And this whole project was a joint effort of the two laboratories of Philip Kokura in Oxford and Petra Schwille here in Munich. So all biological processes are mediated by molecular interactions, and one of the a very prominent example of this, which we have heard a lot about recently, is the interaction between an antibody and its antigen, for example. This interaction has to be mediated, has to be tuned very finely. This interaction has to be tuned very carefully, so the body recognizes structures that are foreign to it in order to mark potential targets for the immune system. In a cellular context, we also encounter very often self-assembly processes of proteins, like we can see in this example shown in the picture here with the actin cytoskeleton. Now, it is not so easy it is not so easy and technically challenging sometimes to investigate these interactions because we can either use because we can either use let me start again. Because we can either use bulk techniques in order to look at averaged average properties of the proteins at hand, or if we use single molecule techniques, then we need to often label the proteins in order to see them. Now, one way of investigating these interactions on a single molecule level, completely label free, is mass photometry, which probably most of the audience today has heard about. With this technique, you use a microscope to direct light onto a sample, which is composed of a cover slip that holds a drop of protein solution. And with the same microscope, you collect the light that returns from the sample. And this light is a combination of two things. So one is the reflected light from the water-glass interface. And the second component is the scattered light from the molecules that land on the surface. And these two lights interfere with each other on the camera, such that the proteins appear as dark spots on a bright background. So if you record a movie of the landing molecules, you see these dark spots. And the special property of this technique is that the darkness of these spots corresponds to the size of the objects that you're observing. So for each of these landing events, you can determine the amplitude of the signal and relate it to the size of the molecule. So if you do that for a number of different proteins that you know the exact size of, you can calibrate your signal and directly relate it to molecular mass of the proteins that you're measuring. With this, now you can use this to investigate unknown protein systems. For example, this has been done to study the interaction between an antibody and an IgG antibody and an Fc gamma receptor in a one-shot measurement. And also we've used this to study oligomerization reactions, like shown here, for actin down to the level of individual actin subunits and small seeds. Now this is all nice, and you can use it for a lot of different protein systems, but at the moment we can only use it for soluble ones. Now next, after we've developed this, we thought, what can we do with proteins that are associated with cellular membranes? Because a lot of the interactions in cells actually take place at the interface between the cytosol and the plasma membrane, or on the plasma membrane itself. Now, in order to do that, we need to first consider a general problem when we do label-free single molecule imaging based on light scattering. So basically all things scatter light, and therefore if you in fact look through a mass photometer on the glass surface itself, you see mostly the pattern of the glass surface roughness, which has approximately 100-fold stronger signal than the proteins themselves. So if you want to look at a protein in this scenario, it's a bit as if you would look at a very tiny stone that you try to make out in a mountain landscape. So if I now throw a little stone into to the bushes next to these rocks here, it's really hard to make out this event or actually look at the details of the event. But you can apply an image processing trick. If I just remove everything from the image that is there before the stone hit the image, then in the differential image, it's really easy to see this spot afterwards. And this is exactly what we use to detect the molecules in mass photometry. So we remove the static background from the glass surface roughness and watch all and watch all the molecule lands afterwards. Now, of course, if we only do this once, since we don't only throw one stone, but basically many on the surface gets crowded very quickly, and the whole and the background also gets blurry because of stage drift. To circumvent this, mass photometry uses a continuous background renewal strategy, where you basically continuously renew the background that you take, and as a molecule lands on a surface and becomes part of the frames, its signal appears in the differential image. Has a peak contrast at the time when all frames contain the particle, but it is not contained in the background frames. And as this whole background removal window slides further through the frames, the particle becomes part of the background and it is removed again. And this way we can separate all the landing events of the molecules clearly and determine their contrast. Now, if we apply the same strategy to a molecule that moves around on a membrane because of diffusion, then we run into the trouble that if the molecule is at this place in the background frame, and then over time moves to a different position, then in the background subtracted video, you will find a lack of particle density at the previous location and additional density where it has moved afterwards. And this causes the moving particles to appear as dark spots carrying bright tails in the differential videos. Now you can circumvent this problem if you apply a completely different background estimation procedure. So one way how you can or what you need to do is basically determine the background without any of the moving particles. So if a particle moves along during a frame sequence, if you look at one particular spot, for example, this here, then you will notice that the signal intensity during most of the frames will not contain the particle. That means if we take the median intensity at this particular position, then the median will be a good estimate of only the background without the particle. Now, of course, this only works if the particle moves sufficiently large distances during the median period. And we can also not take median periods arbitrarily long, because at some point we also run into the trouble of sample drift, and then my lattice doesn't stay where it is, basically. So one of Philip Kukura's former PhD students, Gavin, came up with a really nice idea to actually calculate the median over a frame sequence around a central frame that we are interested in, where the median window is long enough so that that we only estimate the background, and then rather than doing this only once, we shift this median window along with the frames that we are looking at, and this way we always have a refreshed background, but always an estimate of only the static glass surface roughness. And if we apply this image processing strategy to the video that I showed you before, we get a very clear video of the moving particle without any distortions of the point spread function at all. Now we can go back and use our mass photometry detection machinery and determine the contrast of these particles in each frame. And what we get is a time-resolved trace of the mass of the particle as it changes over time. Now, of course, for one particular protein, this mass won't change much. So we can take this kind of the central value for the contrast to get a very accurate estimation of its mass. But what the additional information that we get now from this experiment is also the location of the particle in each frame, and we can actually track this into a trajectory and follow the particle's movement. And doing this, we can apply common approaches and basically extract diffusion properties like the diffusion coefficient from the trajectory. So Eric and Manish from Philip Kukura's lab have done this and used this approach to investigate a protein called dynamin, which is known to form oligomers on lipid bilayers, and they have tracked many of these particles, and basically for all of these particles determined both the mass and the diffusion coefficient, and plotted these two characteristics in a 2D map that is shown here. And as you can appreciate, they found the different molecular species as distinct islands in this map. The contrast of these molecules scaled nicely linearly with the expected mass for the oligomers. So it was kind of self-calibrating, and we could easily assign which contrast corresponded to which oligomeric species of the dynamin molecule. But what you can also see is that the different species had different diffusion coefficients, which were inversely proportional to the number of subunits in the complex, which shows that every time a subunit of the dynamin is added to the complex, there are probably more contact sites to the lipid bilayer. From the length of the trajectories that they got, they could also estimate off rates of the complex from the membrane, which also scaled inversely with the number of subunits. Now, with these tools in hand, we thought, okay, let's try this approach on a slightly more complicated biological system. And this was when we teamed up with a group of Petraschwille, who are experts on investigating membrane proteins in vitro. And one of their special systems that they like to look at is the E. coli min system, which in E. coli is used to coordinate the cell division cycle. So there are these two proteins, min-D and min-E, which oscillate between the cell poles of E. coli and basically establish a concentration gradient between the cell poles of a third protein, min-C, which is a passenger protein on these two proteins. And this third protein can then prevent the Z-ring forming protein FTSZ from interacting with the membrane everywhere except where the min-C is not located, and thereby the whole system coordinates the formation of the cytokinetic ring during cell division. Now, these two proteins, MinD and MinE, have very interesting properties. If you actually use the purified proteins and put them on a supported lipid bilayer in vitro, they start to generate these fascinating patterns on the membrane completely by themselves. As you can see here, they move in they form this kind of spiral wave on the bilayer, where MinD in green moves along the wave front and is chased by MinE in red here. And how people think this is accomplished is through this canonical reaction cycle of MinD and MinE. So MinD as a monomer binds ATP, dimerizes, and binds to the bilayer, and then MinE attaches to MinD, triggers the ATPase activity. MinD hydrolyzes ATP, releases its phosphate, and gets released as monomers from the membrane. However, it's not really clear how this simple reaction scheme can account for the complex pattern formation that is observed in these in these waves, for example, because you somehow need some sort of anisotropic distribution of the protein, which you wouldn't expect to get from this simple reaction scheme. So we decided we could use our new technology to investigate this reaction cycle a bit further. First of all, if you don't have a protein like dynamin, which is more or less self-calibrating, you need some sort of independent mass calibration for this new approach. And what we did is we used biotinylated standard proteins that we attached via streptavidin to a biotinylated bilayer. And this way we could generate bilayers with differently sized proteins diffusing on top of it. And then from our 2D maps of diffusion coefficient versus interferometric scattering contrast, we could nicely distinguish in the species that would form and could assign the peaks to the expected masses and generate a calibration line similar to the one that we normally do for mass photometry. Now, with a well-calibrated system, we went on and tried to systematically probe the MinD reaction cycle. So starting with it, we compared the state of MinD in solution as compared to how it assembles on the membrane. And so in solution, we use a conventional mass photometry experiment, showing us a peak at around 33 kilodalton, which corresponds to MinD monomers. And on a sparsely populated membrane, we found that MinD mostly assembled into dimers, which basically confirms the existing model. But then as you increase the MinD concentration in solution, you can also push the MinD density on the membrane. And what we observed there was that the mass distribution shifted towards higher masses that couldn't only be explained by MinD dimers alone. Now, as a control, to see if we can revert this, we used a MinD mutant that was a MinD mutant that was published or described to have a locked dimer interface. That means it can only form a dimer. And when we put that onto the membrane, quite surprisingly, we found that indeed, yes, we see a dimer population for this mutant, but also as the density on the membrane increased, we found that we can also see a clearly distinct peak for MIND tetramer. Now, what we think that means is that apart from the canonical dimerization interface, we have an additional assembly interface that can hold more MIND subunits. And the difference between the mutant and the wild type is that due to the locked dimer interface, we preferentially get dimers and tetramers for the mutant, but we can also have intermediates where only one subunit of MIND attaches for the wild type. Now, so far, we've always just looked at the center mass that we get out from the entire trajectory. As I told you in the beginning, we can for each point in the trajectory, we can determine a mass for our protein and then determine a kind of an average mass. But in fact, since we get a mass estimation for each point in this trajectory, we actually get a time-resolved trace of how the mass changes over time. That means, in principle, we should be able to look at real-time assembly disassembly if it occurs. So for MIND, in most of our trajectories, especially if they were short, we saw a more or less flat line. But in some of the longer trajectories, the mass would fluctuate more than in these flat lines. And we thought this could be due to attachment and detachment events of subunits in between. Now, because the shifts in the mass were very tiny for the MIND system, we used the step detection algorithm that would use statistical criteria to detect steps in the trace. And if we applied this to these two different traces, for example, we can extract a lot more information than just from the single point measurement. For example, here we can determine the size of subunits that are being attached or that are being detached, and we can very accurately determine the size of subunits or the complexes, how they are being completely released from the membrane as the trajectory ends. So if we use the step size distribution that from the steps that we've detected, we can see that both for attachment and detachment events, we get a distribution centered around dimers for low particle densities. So in this graph, light to dark means with increasing protein densities on the bilayer. And so if we increase the concentration of, or the crowdedness of the bilayer, you can appreciate that the whole, also the complexes that are attaching and detaching from our molecules are getting bigger. Because we can also now determine from our step analysis where the exact what the exact mass of our proteins is when they get released from the membrane, and we can determine that mass much more precisely rather than just having the blur of like the whole trajectory of the mass over time. We can also narrow down the mass distributions of complexes that are being released from the membrane, and you can appreciate that now the MIND wild type actually can attach in either dimers, trimers, or tetramers. So indeed, it seems to actually have the is able to form these species. For comparison, the locked dimer mutant can only detach from the membrane as a dimer or tetramer. So now, from the length of the plateaus in the step in the step traces, we can also estimate lifetimes of the complexes on the membrane. We can, for example, determine on and off rates from plateaus before such events, or we can determine the release rate of complexes that are completely dissociating from the membrane. And basically, to summarize, here light to dark means larger oligomers. So in general, larger oligomers are also more stable on the membrane. So basically, we need to modify the canonical model of MIN-D assembly on the membrane. And what we've learned is that, yes, MIN-D assembles from monomers as dimers onto the membrane, but then it can also recruit MIN-D subunits from the solution and grow larger, even up to the size of a tetramer. So, and what we finally did, we also wanted to see what happens when we add MIN-D to this system. So, looking at the same size distributions with MIN-E, we found that MIN-E would actually stabilize a tetrameric species, and if you increase the density of MIN-DE on the membrane, the complexes would grow extremely large, even larger than the size of MIN-D alone. And if we also apply our time-resolved analysis to the traces of MIN-DE, you can see again that the higher the protein density on the membrane gets, the larger objects actually attach and detach to our complexes, and the complexes that are being released from the membrane are getting really, really large, up to something corresponding to MIN-D 8-mers or 10-mers as the protein density on the bilayer is high. Also, the lifetimes of MIN-DE species is always higher as compared to the corresponding MIN-D alone. So what that means is that it seems like when MIN-E binds MIN-D, it doesn't immediately lead to its dissociation, but it can rather stabilize the protein on the membrane and can lead to a bridging effect between subunits of MIN-D, which can lead to the formation of really large oligomers of MIN-D and E. And these oligomers can then, in a concerted manner, be released from the membrane, and we think that these complexes might even be transiently stable in solution, so that they can rebind, and this might contribute to the accumulation of MIN-D, or the local accumulation of MIN-D as it's observed in these patterns on the membrane. So I hope I could convince you that mass-sensitive particle tracking is quite a powerful new tool to investigate the mechanistic details of how proteins interact with membrane and with themselves on the membrane, and that this can be a really powerful new technique to reveal structure-function relationships on lipid for proteins that are diffusing on lipid bilayers. Finally, I would like to thank everybody who's participated in this study, both Petra Schwiller's lab and also my old lab in Oxford. It has been a great pleasure to work with you, and really fun. And at this point, I would also like to point out that if you want to learn more about these about this technique, you can look on our bioRxiv preprints. We are currently trying to publish both the MIN data and the Dynamin data back to back. Yeah, and I would like to thank you for your attention, and I'm happy to take questions.
Thank you for your presentation. As a reminder to our audience, you may submit your questions to the speakers using the Q&A tab at any time. Our audience has already submitted several questions, so let's get to them. Joining Dr. Hund are Tamara Herrmann and Frederick Steyert from the Laboratory of Peterschwille at the Max Planck Institute of Biochemistry. Welcome. So, I guess to start, can this be used for integral membrane or transmembrane proteins? And have you tested proteins in nanodiscs or micelles with this technique?
Yeah, maybe I can start to answer this question. So maybe I start with the second question. So mass photometry has been used for integral membrane proteins with nanodiscs, but in that case, you can actually do the simple landing assay as is normally used in mass photometry experiments, because basically what you do is you solubilize your integral membrane proteins, and then they land as normal soluble proteins on your surface, and you can determine interactions with it. But with this particular technique here, we haven't done integral membrane proteins yet. I suppose it is probably possible. It's just you need to change the way you make the bilayer, because usually you need something like a cushioned bilayer to do that.
Great, thank you. So some proteins make higher order oligomers at higher concentrations, and it seems that using high concentrations of protein in this technique could lead to a crowding effect. Is that so? Can you speak to that?
Yeah, I mean, of course, the higher the higher the protein concentration in solution, the more proteins also are attached to the membrane, and then of course the membrane becomes more crowded. So what you need to make sure is that your membrane doesn't get so crowded that you can't distinguish individual molecules anymore in your image. But in general, I think the crowding effect on the bilayer definitely will help the proteins to find each other and increase the size of the oligomers. Does that answer your question?
Yes, thank you. What are the lower mass detection limit and mass resolution for diffusing molecules on the SLB?
Yeah, so in principle, it's very similar to conventional mass photometry. So maybe slightly worse. It depends really on the imaging conditions and also on the size of the molecules that you're working with. So let's say, like, for example, for a protein in the range of like 60 kilodalton, such as the MinD dimer, we observed something like 30 kilodalton full width half maximum resolution, which is, I think, very close to the mass photometry resolution. And also the lower mass detection limit, again, depends a little bit on your imaging conditions. So we have used a fairly small field of view, which was like 9 by 3 micron or so, and basically used the refine instrument's maximum frame rate for that. So the effective frame rate at the end was, I think, 200 frames per second, and with that we could go down to proteins that were about 50 kilodaltons in size. That was the lowest mass.
Great, thank you. This next question pertains to the lipids. How exactly are they spread? Like, what is the technique? And are there any lipid species that are either not suitable for the technique or any limitations to what the composition can be?
Tamara, do you want to say something on that?
Yeah, sure. So we actually do supported lipid bilayers, in this case, kind of conventionally by vesicle rupturing. So what is kind of important in this case is to extrude the vesicles to have a monodisperse vesicle mixture to kind of ensure that your supported lipid bilayer in the end is really integral, to kind of have good diffusion properties. I guess most lipid mixtures should be fine. The minute you can make a normal supported lipid bilayer for example, fluorescence measurements or something, you can definitely also do this for MSPT. But of course, there are kind of tricks to some lipids, like for example, PIB, where you kind of need to ensure, like for example, protonation just before adding them to the mixture so that they kind of incorporate nicely. But in principle, every lipid mixture should be possible.
Great, thank you. Is it possible to use MP landing assay calibrations for diffusing molecules?
I can go with that question. So we recommend to do not, because I mean, of course, you as you've seen in one of my slides, the calibration lines of the of mass photometry and MSPT on the same instrument fell very nicely on top of each other. But we rather think that this is a coincidence, because we've observed that depending on your imaging conditions, like how fast you image, and also what kind of buffer lipid combination you use, this might change slightly. So we recommend to always do a calibration for the system that you're measuring at.
Great, thank you. We've received several questions about the experiment itself, the MSPT experiment, but how much time is needed for the various steps, like preparation, how much time for the measurement, analysis, and so forth?
Yeah, I think I can answer this. So, I mean, preparing kind of the time-consuming steps kind of in the beginning would be like cleaning the glass cover slides and, for example, building flow chambers, but there are, of course, also commercially available flow chambers that also should work. Vesicle preparation also takes some time, so I would estimate roughly until you have, until you kind of really can start on the microscope, it would be something between 2 and 3 hours. And then our measurement times kind of were roughly 6 minutes per measurement on an SLB. But for example, you need some time to actually also prepare the SLB, so that's another 10 minutes, I would estimate. But of course, the measurement time itself depends on your system, so kind of on what time scales your system of interest is actually performing. So for the MIN system, it's actually quite fast. And for the data analysis, actually, I would roughly estimate also another like 2 or 3 hours to actually kind of, get all the analysis steps on. Yeah.
Great, thank you. Is there a way to collect or enrich for proteins that you're interested in after or during this analysis, like monomers of multimers of a certain size for any follow-up analysis?
Yeah, I don't know, Frederick, do you want to say something about that?
Yeah, I'm not sure if I understand this correctly, but I mean, you can, of course, filter your data set by mass, or I don't know in which direction this question goes.
Yeah, I think it's asking if there's a way to actually collect the proteins from the slide to do further analysis if they are multimerizing.
Yeah, so I mean, I would say so of course, like in the assay itself, it's really hard to select for certain oligomers. But of course, as Frederick said, we can, depending on how well your protein species are separated, of course, by mass. I mean, for example, in the Dynamin case, you've seen that you can clearly distinguish the individual species as different peaks. So you can, for each protein, very accurately assign what kind of molecule this might be. So in that case, of course, you can filter your data set after during the analysis and only look at the properties of one particular oligomer species. I think that's actually the power of the technique.
Thank you. Is it possible to measure or look at drug interactions with your membrane proteins using this system?
I mean, I guess so. Drugs are normally quite small, so visualizing the drug itself would be, I guess, hard. But if your drug, for example, induces oligomerization, you should definitely see the oligomerization that is being induced.
Thank you. We may have already touched on this slightly, but what time scales in terms of frame rates and measurement lengths does this operate on?
Basically, it's the same time scales and frame rates that you can achieve with the mass photometry instrument that you have. So, for example, in our case, we used one of the commercial refined setups, and in the small field of view, this setup I think can achieve frame rates up to 1 kilohertz, and the field of view size in that setting is about 9×3 micron. So you can go extremely fast with it if you want, but of course you can also go to larger field of views if you don't have to resolve really small proteins or detect really small proteins. Then your field of view size might be larger to some more something like maybe 20×20 micron or something. But then also your frame rate goes down. So usually it's basically it's restricted by the amount of light that you can direct onto your sample, that your sample can tolerate, and also that the camera can handle, basically.
Thank you. Is it possible to distinguish subunit assembly events that occur on the membrane versus those occurring in solution?
Yeah, that's a good question. So, I mean, that really, I mean, it depends on your system really what is more likely to happen. I mean, if you, for example, have a system like in our case, the MIN system, we think that most of the events where subunits attached to complexes on the membrane came from solution. But yeah, maybe you can switch over to my screen again. I have a nice video that Manish and Eric have taken from for the Dynamin case. Is my screen shared?
Yeah,
okay.
If you just want to go into full screen mode.
Yeah. So basically, they have looked into they have found quite rare cases for their example where they show a dissociation event, like you can see this central particle is moving around and then it splits into two. So this does happen, and you can watch it. And also sometimes what happens, you can if you see the particles, they actually merge into a bigger particle like this. I mean, these events have been really rare to observe in their case, and it's also like software wise, we are not able to quantify or detect these events yet, because probably the tracking program would in this case kind of stop the trajectory and start a new particle or something. So we would have to probably adjust the image processing and particle analysis software to account for these events.
Great, thank you. Are there any steps in the protocol that require special care or attention?
Yeah, I mean, it all kind of comes down to having a really good membrane. So having a kind of diffusive membrane is really key kind of to these experiments, and of course the background. So we would highly recommend that all buffers need to be filtered prior to their use so that you kind of don't get a high background noise. And of course, everything that you additionally add that could kind of aggregate in your buffer really needs to be taken care of, because that would obscure the background.
Great, thank you. Could you comment on now analyzing proteins that may deform the membrane or induce curvature, like, for example, perhaps the dynamin in your data sets? Do you see anything like that?
Yeah, I mean, this is hard because, I mean, we are working on supported bilayers, so I guess it's quite hard for the proteins to induce curvature. If at all, they would probably just locally make a slight curvature or something, which is hard to detect in our system because, of course, all these things are still detected with optics for light microscopy, and they are all diffraction limited. So I would not expect that you would see any effects with like small scale membrane curvature or so. At least we haven't seen that really.
Great, thank you. In the microscopy setup, is there any benefit to changing or scanning the axial position of the focal plane, or is the best signal always obtained when the focus is exactly at the lipid bilayer?
Yeah, I mean, yeah. So in iSCAD microscopy, it's a little bit, it's kind of a yeah. I mean, the way we define the focus is that it's the focal plane where the objects that we are looking at have the highest contrast, basically. So in a way, of course, like if you change the axial position, the way the setup is aligned, you get the clearest and crispest image when you actually are in the focal plane. I mean, of course, there are publications where people use the kind of 3D distribution of the point spread function to more accurately determine the position of a molecule. But this is more advanced, and we haven't really taken or used this for our analysis approach.
Great, thank you. And now we have time for just one more question. How do you determine MIND monomers in the solution, since it seems like their size may be close to the lower mass detection limit of the technique, depending on background noise?
Yeah, so yeah, that's a good question. So for the solution experiments, we have actually used a non-commercial setup, which was slightly more sensitive than the one from Refine. It had a blue laser, which generates a stronger scattering signal, basically, and the sensitivity of that setup was higher than for the one from Refine. So we could go down to these really small proteins and still detect them. Maybe not quantitatively, but at least we could detect them. Of course, like on the membrane, we cannot completely rule out that there's also MIND monomers, and we think that there actually might be. The problem is at our imaging sensitivity, we weren't able to detect the MIND monomers on the membrane because, as you say correctly, the lower mass detection limit is too high for these species.
Great, thank you. And I actually had one more come in. How do you get sub-diffraction limit resolution to resolve protein particles in the X, Y, and Z?
Yeah, so should I answer it, or do you want?
Yeah, go ahead.
So this works like in any other fluorescence-based super resolution technique that you fit a point spread function to your spot detection. So you get subdifraction resolution, but not in Z because it's planar.
Great, thank you. Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the Scientist, I'd also like to thank our speaker, Dr. Nicholas Hund, our Q&A participants, Tamara Herrmann and Frederick Steyert, as well as our sponsor, Refine. Thanks, everyone, and goodbye.